Okay, so myself, Dr. K. Shivaya, Department of Pharmacology and TVM College of Pharmacy. So, in the continuous of the last class, we can discuss about TCA cycle, tricarboxylic acid cycle. This also we can call as a Krebs cycle. So, why it is called Krebs cycle? The scientist name is so the Krebs scientist these investigated and discovered the tricarboxylic acid cycle, and then also it is called as citric acid cycle. So in the first step, it formed the citric acid. That's why it is called as citric acid cycle. Okay. Last class, already we understood the glycolysis process. In the glycolysis process, the glucose is converted into pyruvate. So these pyruvate, after that, how it is utilized in our cells, how it is metabolized, or means nothing but catalyzed. Okay, so these pyruvate, when the transamination takes place, transamination means addition of amine group to the pyruvate. So when the transamination takes place, it produces the alanine and pyruvate. When the from the pyruvate carboxylation takes place, carboxylation reaction happen to produce the oxaloacetate. Then decarboxylation, the removal of carbon dioxide, the removal of carbon dioxide from the pyruvate, that it produces the acetyl coenzyme A. It's required, it is thus acetyl coenzyme A, it is enter into the citric acid cycle, we can discuss later. Then the pyruvate reduction means addition of hydrogen molecules and in the presence of lactate dehydrogenase enzyme. The pyruvate is converted into lactate in the presence of lactate dehydrogenase enzyme. It is happen the deficiency of oxygen, anaerobic glycolysis. So the pyruvate is converted into lactates. So this is the how the pyruvate metabolizes different products. Maximum the pyruvate it is enter into oxidative decarboxylation and enter into citric acid cycle. So the link between the glycolysis and tricarboxyl cycle is the link is the pyruvate to convert into acetyl coenzyme A in the presence of pyruvate dehydrogenase. Pyruvate dehydrogenase. I said already the glycolysis process where it is happen, it is in the cytosol. It is in the cytosol. Then the pyruvate enter into the mitochondria. These pyruvate, in the presence of pyruvate dehydrogenase enzyme, it is converted into this is oxidative decarboxylation. This process we can call as decarbylation, the removal of carbon dioxide present in the pyruvate and to form the acetyl coenzyme A. And to form the acetyl coenzyme A. These pyruvate dehydrogenase enzyme have the complex. It's required 5 coenzymes. That the 5 coenzymes are TPP, thiamine pyrophosphate, TPP, thiamine pyrophosphate, lipoamide, lipoic acid combined with the lysine amino acid. That's why it is called as lipoamide. Next, FAD, flavin adenine dinucleotide. Flavin, F means flavin, A means adenine, D means dinucleotide, N means nicotinamide, adenine dinucleotide, and coenzyme. These coenzymes combined with the PDH pyruvate dehydrogenase enzyme and to form the acetyl coenzyme A. Without these five coenzymes, the acetyl coenzyme it is not formed. During the in the formation of acetyl coenzyme A, it lost the carbon dioxide. So that is a decarboxylation. So the citric acid cycle, it is mitochondria. The citric acid cycle is a center pathway. Why it is called the central pathway? The carbohydrates enter into the finally the citric acid cycle, amino acids and lipids. Amino acids and lipids finally enter into the citric acid pathway. Okay, that's why it is a central pathway for all carbohydrates, amino acids, and lipids. So finally, to form the carbon dioxide, it releases the carbon dioxide and finally to produce the energy. So here, how? So in the glycogenolysis, when the glycogenolysis takes place, glucose formation, the glycolysis of pyruvate is formed. The oxidative decarboxylation, acetyl coenzyme. Suppose if you take in triglycerides means lipolysis, lipids, free fatty acids, beta oxidation. After beta oxidation to form the acetyl coenzyme, then protein form the amino acids. When the deamination happen, deamination reaction, when it is takes place and oxidation takes place to form the acetyl coenzyme. That's why the citric acid cycle, the final common pathway for the carbohydrate, lipids, and protein. Come to the citric acid cycle pathway. So in this pathway, first the formation of citrate, formation of citrate or citric acid. That's why it is called citric acid cycle. I said already the pyruvate is converted into acetyl coenzyme A. The pyruvate is converted into acetyl coenzyme A. It is combined with the OAA, means oxaloacetate. Oxaloacetate. Oxaloacetate to form the citrate. Form the citrate. Then after that, the formation of citrate is irreversible. The formation of citrate is irreversible. Then these citrate in the presence of aconitase enzyme, the presence of aconitase enzyme, in the presence of here it is very easy to understand. In the presence of aconitase enzyme to form the isocitrate, to form the isocitrate. Then these isocitrate is converted into alpha ketoglutarate in the presence of isocitrate dehydrogenase enzyme. Remember, when the dehydrogenase enzyme came in the reaction, it lost the one hydrogen molecule. It lost the hydrogen molecule from the substrate. That hydrogen molecule to form the NADH H plus. The NAD plus converted into NADH plus H plus. Here it is lost the carbon dioxide. It lost the carbon dioxide and to form the alpha ketoglutarate. Alpha ketoglutarate. It is first just brief. Then alpha ketoglutarate in the presence of alpha ketoglutarate dehydrogenase enzyme. Here also dehydrogenase enzyme comes so that it lost the substrate lost the hydrogen molecule. So the hydrogen molecule added to the NAD plus to form the NADH H plus. So these the alpha ketoglutarate to form the succinyl coenzyme A. Succinyl coenzyme A. The succinyl coenzyme A, the presence of succinate thiokinase. Succinate thiokinase. I said that when the kinase enzyme came, either phosphorylation, either dephosphorylation happened. So here the GDP, guanosine diphosphate, GDP means guanosine diphosphate to form the GTP. From the GTP, it lost the one phosphate group from the succinate coenzyme to form the GTP. This is a substrate level phosphorylation. How in the glycolysis, glycerol head 1,3 bisphosphate is formed to the glycerol 3 phosphate. How it is formed? Like this also, succinate coenzyme, it is converted into succinate in the presence of succinate thiokinase. Succinate thiokinase to form the succinate. Then succinate again to form the fumarate in the presence of succinate dehydrogenase. When the dehydrogenase enzyme came, lost the hydrogen molecule from this substrate here, succinate. So the FAD added the H plus, so FADH2 formed. So then the succinate to form converted into fumarate in presence of succinate dehydrogenase enzyme. So these fumarate in the presence of the fumarase enzyme to form the malate structure. Then the malate in the presence of malate dehydrogenase, I said already, when the dehydrogenase enzyme came, here the loss of hydrogen molecule from the substrate. So the NAD plus is converted into NADH, H plus plus H plus. Then the malate is converted into oxaloacetate. These oxaloacetate again, it is involved in next acetyl coenzyme A. Again, it bind to the acetyl coenzyme A again to form the citrate in the presence of citrate synthetase. Here we can elaborate the reactions, the citric acid cycle. Okay, it is present in Satyanarayana textbook. Here we can they presented with the structure. See, this is a pyruvate. This is a pyruvate enol form, not keto form. It is enol form. So these keto form, these keto form. Sorry, not keto form. These pyruvate keto form. I said to form the acetyl coenzyme in the presence of pyruvate dehydrogenase enzyme. Just remember the structure: CH3 C double bond O C O minus. So in the presence of these coenzymes, I said 5 coenzymes: FAD, NADH plus, coenzyme A, uh, and copamide and TPP. So these are all involved in the formation of the acetyl coenzyme. And then the acetyl coenzyme combined with the oxaloacetate. I said already OXA. So this is oxaloacetate. It is end product of the citric acid cycle. It is bind with the acetyl coenzyme to form the citrate. See, just oxaloacetate and acetyl coenzyme, remember structures. We can write here. If you first, you have to write the all names followed by, if you have time in the examination, you write the structure. Otherwise, you leave it. So citrate synthetase to form the citric acid. So that's why C, CH2COO, CH3OH, and COO, CH2COO. Then in the presence of aconitate, it lost the carbon, hydrogen molecule, water molecule, it lost the water molecule to form the aconitis. Here it is, we can write these aconite, or otherwise you can go directly, you can write isocitrate, no problem. So in the presence of iconitase enzyme to form the aconite, it lost, then again it will formed. Then isocitrate, it is formed. Okay. So here the formation of citrate, the citric acid cycle is irreversible. Irreversible means the citrate again, it cannot form oxaloacetate and acetyl coenzyme. Fine. The isocitrate in the presence of isocitrated dehydrogenase. So here lost the 1H plus in the isocitrate hydrogen molecule. Here the hydrogen molecule is lost. Here just to observe the, okay, to form the oxalosuccinate. That hydrogen molecule utilized by the NADH plus. Next, then oxaloacetate to form the alpha ketoglutarate in the presence of uh, sorry, it is oxalosuccinate dehydrogenase enzyme. Dehydrogenase enzyme. Same enzyme, same isocitrate dehydrogenase enzyme to form the alpha ketoglutarate. Isocitrate dehydrogenase to form the alpha ketoglutarate. Okay, so here in the intermediate product is oxalosuccinate. Oxalosuccinate. Then you have to write this isocitrate, and directly we can write alpha ketoglutarate in the presence of isocitrate dehydrogenase enzyme, and you have to write in the bracket compulsory NAD plus NADH plus and carbon dioxide removal. Okay, then alpha ketoglutarate. These alpha ketoglutarate to form the succinyl coenzyme A in the presence of water alpha ketoglutarate dehydrogenase. Here the again the removal of hydrogen molecules, and here the coenzyme A involved, So that's why to form the succinyl coenzyme A, succinyl coenzyme A. So that the H plus molecules utilized by the NAD plus are to form the NADH plus H plus. NAD plus utilize the H plus H molecules and NAD to form the NADH plus H plus. So then succinyl coenzyme in the presence of succinate thiokinase. Here the thiokinase. So to form the succinate, here write directly name. If you remember the structure, you write it. Otherwise, you write the direct name. Then succinate, the succinate in the presence of succinate dehydrogenase to form the fumarate. So here the dehydrogenase, it loses the hydrogen molecules. It loses the hydrogen molecules, and that lost the hydrogen molecules utilized the FADH2. Okay, form the FADH2. Then fumarate is converted into malate in the presence of fumarase. Here it lost the hydrogen molecule, water molecule loss. Then malate is to form the oxaloacetate in the presence of malate dehydrogenase enzyme. Then oxaloacetate again enter into the citric acid cycle in the synthesis of citric. This is a pathway of citric acid cycle. Here we have to observe the important steps are, that is irreversible reactions that are, see, I given that dark mark that it only one side, not a double side. Here the double side marking is there. Here the dark mark only one single side is there. Here the formation of citrate is only takes place. It does not form the oxaloacetal coenzyme. So like that, same it is isocitrate to alpha ketoglutarate. Isocitrate to isocitrate to alpha ketoglutarate also does not come back. Means alpha ketoglutarate does not come back to form the oxalosuccinate and does not form the isocitrate. So the once it is formed alpha ketoglutarate, it does not form the isocitrate. I give it, this is the one side only, that thick. And the alpha ketoglutarate, when it is formed, it's succinyl coenzyme. When it is formed in the presence of alpha ketoglutarate dehydrogenase enzyme, it again is one more irreversible reaction. Reversible reaction means here I'm saying citrate is converted into aconitate, and then aconitate again to form the citrate. That's why double arrow marking here in the presence of aconitate enzyme. That is a reversible reaction. Irreversible means irreversible. It will not come back. So here the succinyl coenzyme is converted into succinate. This is also a reversible reaction. So these one and for these four reactions, irreversible reactions present in the citric acid cycle, and these enzymes, that is that is citrate synthetase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and succinate thiokinase enzymes are to regulate the citric acid cycle, to regulate the citric acid cycle. This is regarding the citric acid cycle. Then come to the energetics, where the already you studied the NADH, FADH, and the GTPs are to produce the energy. So here in the formation of isocitrate to alpha ketoglutarate, what NADH H plus is formed. Again, alpha ketoglutarate to succussion coenzyme, second NADH plus H plus is formed. Then again, third one NADH is malate to oxaloacetate, malate to oxalate. Here one more. So total 3 NADH plus H plus are formed. And next one FADH formed, FADH2 from in the during the formation of succinate to fumarate, it is formed. So succinate to fumarate, one FADH2 formed, and one GTP also formed. So this is for the one acetyl coenzyme if it is entered. But in glycolysis, two pyruvates formed because of after splitting glyceraldehyde 3-phosphate and dihydroester phosphate. The glyceraldehyde phosphate to form the pyruvate. Again, the dihydroester phosphate is converted to glyceraldehyde 3-phosphate, and one more pyruvate is formed. Already we discussed in glycolysis. So finally, in the glycolysis, two pyruvates form. When the two pyruvates form, two acetyl coenzymes are formed. So then you can get the 6 NADH. So here the 3 NADH plus H plus is there. So you can get the 2 into 3. So because of two acetyl coenzymes and also 6 NADH plus H plus, one glucose molecule per, and 2 GTPs, 2 FADH. Okay. Then significance of this citric acid cycle, and how it is utilized in the citric acid cycle, the intermediate metabolites, how it is utilized. See, so this is first we can talk about energy levels. So 3 NADH in a total turn off cycle, 3 NADH means 9 ATPs, 1 FADH2 from 1 FE. See, 1 NADH H plus 1 NADH plus H plus to produces the 3 ATPs. 1 FADH2 produces 2 ATPs. So for one acetyl coenzyme 3 NADH H plus, so 3 into 3. So because of 1 NADH H plus produces 3 ATPs. So that's why 3 into 3, 9 ATPs. 1 FADH produces 2 ATPs, that is 2 ATPs. So 1 GTP is formed, 1 ATP. Total is 12. This is for the one acetyl coenzyme A. This is for one acetyl coenzyme A. But actually, two acetyl coenzyme A it is formed. That's why, so 2 pyruvates is formed because of 2 pyruvates formed in the glycolysis. So 2 pyruvates, that's why 12 into 2. 12 into 2. So here it is given 2 acetyl coenzyme A. So 24 ATPs. So in glycolysis at the end, 2 pyruvates formed. You can get the 8 ATPs only. Already we calculated last class. 2 pyruvates is converted into acetyl coenzyme A. So 2 pyruvates, because of 2 pyruvates formed, then after that pyruvate is to form the acetyl coenzyme. It is intermediate step, already we discussed. So here 1 NADH. So 2 pyruvates, that's why. So 2 into 3. 1 NADH plus H plus produces 3 ATPs, 6 ATPs. So 2 acetyl coenzyme A. So in the citric acid cycle, 24 ATP. So like that, we can get the 38 ATPs for one glucose molecule. So one glucose molecule after completion of glycolysis and citric acid cycle to produce the how many ATPs? So 38 ATPs. 38 86 ATPs. This is the energetic yield. The significance and how these citric acid cycle to help in the biological systems, how it is formed other metabolites, how it is formed other synthesis, biosynthesis products. See these acetyl coenzyme, in the first we can get citrate. These citrate useful in the synthesis of cholesterol and fatty acids. Fatty acids formation and cholesterol synthesis. Then isocitrate is formed. Then alpha ketoglutarate. These alpha ketoglutarate useful in the formation of amino acids. It's a cursor, the precursors for the synthesis of amino acids. Next, succinyl coenzyme A. It is useful in the formation of isoleucine amino acid, methionine amino acid, valine amino acid. Some succinyl coenzyme is also useful for the synthesis of some chain in the fatty acid synthesis. Some fatty acid synthesis. Then succinate, then fumarate. These fumarate in the presence of some enzymes to form the aspartate amino acid, phenylalanine amino acid, tyrosine amino acid. So these are all amino acids. And malate, from the malate we can get the glucose molecule in the presence of some enzymes. The oxaloacetate also it is useful in the synthesis of amino acids. If the double mark is there, from the amino acids we can get the oxaloacetate. It's a reversible reaction. Okay. So how these in the citric acid cycle, how it is formed, there's some amino acids, some fatty acids, how it is involved in the biological system. So that is the regarding class of citric acid cycle and Krebs cycle. Okay, thank you.